morning, class! I hope you're all ready for another exciting lesson today. I hope that everyone is alive, alert, and enthusiastic in today's lesson. Today, we're going to dive deep into the fascinating world of lipids. By the end of our session, you'll be able to classify lipids based on their structure, identify the building blocks of saponifiable lipids, understand the formation and roles of triglycerides and phospholipids, recognize the unique structure and functions of steroids and waxes, and finally, comprehend the chemical process of saponification and the crucial role lipids play in our health and nutrition. Before we get into the nitty-gritty of lipid classification, let's jog our memory a bit. Last time, we briefly touched on what lipids do in our bodies. Can anyone remind me, or do you still remember some key biological functions of lipids? Great recall! That's excellent for understanding and remembering that some lipids are essential in the cell membrane. Now, to solidify that understanding, I have a quick matching activity for you on the board. You'll see a list of lipid types and biological functions. Your task is to match each lipid type to its biological function based on the previous discussion. Are you ready? I'll give you 10 seconds. Alright, let's see how well you did. Okay, for the first lipid type, for triglycerides, the correct answer is, of course, it's energy storage. Next, for phospholipids, it's membrane structure. And for steroids, it's very essential for chemical signaling. Perfect! You've done well, and you've remembered the last discussion clearly. Now, since we have already discussed what makes a lipid and its function work together, what do you think makes each lipid different from each other? How do you think they are classified? Now that we know what lipids do in the body, what do you think makes one different from each other? What's the basis for their distinction? Today, we are going to answer that question by looking at their structures. Lipids are an essential group of biomolecules, and they're classified based on their chemical structure and properties. We can broadly divide them into two major categories: simple lipids and complex lipids. Let's start with simple lipids. These are essentially esters of fatty acids with alcohols. For example, fats are formed from glycerol and fatty acids. And when these fats are liquid at room temperature, we call them oils. Then we have waxes, which are formed from fatty acids combined with long-chain alcohols. Moving on to complex lipids. As the name suggests, these are esters of fatty acids that contain additional groups. Think of extra components like phosphate, carbohydrates, or even proteins. Examples include phospholipids, glycolipids, which have a carbohydrate component, and lipoproteins, which are complexes with proteins. Beyond simple and complex, there's another crucial way to classify lipids: saponifiable and non-saponifiable lipids. So, saponifiable lipids are those that contain ester bonds. This means that they can be hydrolyzed or broken down by a base, an acid, or an enzyme. Examples here include fats, oils, waxes, and phospholipids. On the other hand, non-saponifiable lipids lack these ester bonds and therefore cannot be hydrolyzed. Examples include steroids, prostaglandins, and even terpenes. Now that we've seen how lipids are classified, let's start with their essential building blocks, the fatty acids. Fatty acids are carboxylic acids that are used to build many saponifiable lipids, such as triglycerides, phospholipids, and waxes. Their structure is quite interesting. They have two distinct parts. First, there's a polar hydrophilic carboxyl group. That's the head that loves water. And second, there's a hydrophobic hydrocarbon nonpolar chain. That's the tail that avoids water. The dual nature of this molecule, having a water-loving and a water-fearing end, makes them an amphipathic molecule. In biological systems, most fatty acids have an even number of carbon atoms and are unbranched. Depending on the bonds between the carbon atoms, they're classified as either saturated or unsaturated. Saturated fatty acids have only single bonds between their carbon atoms. This makes their hydrocarbon chain straight, allowing them to pack tightly together. This is why they are usually solid at room temperature. Think of butter or animal fat. In contrast, unsaturated fatty acids have one or more double bonds. These double bonds can either be cis, which causes a bend or kink in the chain, or trans, which keeps the chain straight, similar to saturated fatty acids. Checkpoint: what is the primary difference between saturated and unsaturated fatty acids? That's right, the presence of double bonds. This difference in shape is incredibly important. It affects how fatty acids behave in the body and has significant health implications. For instance, the kinks in cis unsaturated fatty acids prevent them from packing as tightly, which is why these oils that are rich in these tend to be liquid at room temperature. It's also important to note that in nature, double bonds in fatty acids are usually in the cis form, and if there are multiple double bonds, they're typically separated by single bonds. They're not conjugated. Now that we understand fatty acids, let's see how they combine to form the most important energy-storing lipids in the body: triglycerides. Triglycerides are also called triacyl glycerols. They are formed when three fatty acids attach to a single glycerol molecule through ester bonds. This reaction is a dehydration process, also known as esterification. It occurs between the glycerol backbone and the fatty acid molecules. For each ester bond formed, a water molecule is removed. So, as you can see here, the red molecule or the red atoms are indicated, which forms water. And the blue hydrocarbon chain or the blue chain from the fatty acids are indicated here so that you could see the ester bonds formed through the esterification reaction. Checkpoint! Can anyone recall the specific chemical process that links glycerol and fatty acids? That's right! The removal of water allows the fatty acids to bond to the glycerol backbone, forming those crucial ester linkages. Because the polar groups are bound in these ester linkages, triglycerides are inherently non-polar and hydrophobic, meaning they are insoluble in water. This is why oil and water don't mix, like in salad dressing. The oil, made mostly of triglycerides, floats on water because of this property. Triglycerides serve as the primary long-term energy storage molecule in our fat cells. When you think about it, when you consume more calories than your body needs, the excess energy is stored as triglycerides. Then, when you exercise or fast or go into diet, these reserves are broken down for energy. This is why regular exercise and balanced eating are so important. They help regulate triglyceride levels in the body, keeping us healthy. To summarize, triglycerides are three esters of fatty acids combined to one single glycerol molecule. After learning how triglycerides store energy, let's move on to a lipid type essential for building cell membranes: phospholipids. Phospholipids are unique lipids because they contain a phosphate group. This creates a molecule with a distinct hydrophilic head or a polar head and a non-polar hydrophobic tails. This unique structure is absolutely critical because it allows phospholipids to spontaneously form bilayers, which makes up the basic framework of all cell membranes. Imagine two layers of phospholipids with their heads facing outwards and towards the water and their tails tucked inside, away from the water. Now, thinking about their structure, can anyone recall what makes the head polar and the tails non-polar in a phospholipid? That's right. The phosphate group makes the molecule hydrophilic, while the hydrocarbon chain makes them largely hydrophilic. Because of this, a phospholipid is also called an amphipathic structure, having both a water-loving and a water-fearing part. This is what makes cell membranes selectively permeable. This is a vital function for all living cells, controlling what enters and leaves. Now that we've seen how phospholipids build membrane, let's explore another essential lipid class, the steroids. Steroids are a distinct group of lipids derived from terpenes. What makes them instantly recognizable is their unique tetracyclic structure. That's 3 fused 6-membered rings and 1 5-membered ring, typically labeled A, B, C, and D. Cholesterol is the most abundant steroid in our bodies. It plays a crucial role in cell membranes, contributing to their fluidity and stability. Beyond that, cholesterol acts as a precursor to many other vital molecules, including steroid hormones, bile salts, and even vitamin D. Yes, it's the same vitamin D that we get from the sun. Speaking of steroid hormones, these include both male and female sex hormones such as testosterone, estradiol, and progesterone. They also include adrenocortical hormones such as aldosterone and cortisone, which regulate various bodily functions. You might also be familiar with steroid drugs like cortisone and prednisolone. These have powerful anti-inflammatory effects and are medically used to treat conditions like asthma and rheumatoid arthritis. Checkpoint! Can anyone recall which type of lipid contains a 4-ring carbon structure? That's right, steroids. So while steroids might not look like the fats we typically think of, they are indeed lipids by nature and play critical regulatory and structural roles in the body. Transitioning from steroids, let's look at another important lipid group, waxes. Waxes are lipid esters made of long-chain fatty acids and monohydric alcohols, typically containing 16 to 36 carbon atoms. Their key characteristic is their protective function in nature, providing waterproofing for plants, animals, and even human skin. Where might you encounter waxes in your daily life? Think about things you touch or use. Perhaps candles or car wax, especially candles. Waxes are very abundant in candles. Exactly. Some common examples of natural waxes include lanolin, which comes from sheep's wool and is used in ointments. Beeswax, produced by bees and used in candles and polishes. Carnauba wax, from Brazilian palm leaves, found in car and floor wax. And even spermaceti wax, from sperm whales used in cosmetics and pharmaceuticals. Most waxes are actually mixtures of esters. Though some may contain hydrocarbons, alcohols, or ketones, their practical uses in waterproofing, polishing, and cosmetics directly linked to their natural protective roles in plants and animals. Now, let's shift gears a bit. What do you think happens when fats or oils like the triglycerides we discussed earlier are heated with a strong base like lye? This brings us to the concept of saponification, a hydrolysis reaction that occurs specifically in an alkaline or basic solution. This process breaks the ester bonds in triglycerides, producing glycerol and fatty acid salts, which we commonly know as soap. It's a two-step process. First, the hydrolysis of the ester bonds in triglycerides forms glycerol and free fatty acids. Then, those fatty acids are neutralized by a strong base, like sodium hydroxide, to produce fatty acid salts, which is our soap and water. Essentially, lipids that don't have these ester bonds are considered non-saponifiable, because there is no avenue for the hydrolysis of these lipids. Now, let's talk about how soap actually cleans. The fatty acid salts formed during saponification have a remarkable property. They form micelles. Micelles are spherical structures where the hydrophilic heads face outwards towards the water, while the hydrophobic tails trap grease and oil inside. This micelle formation is key to how soap effectively removes dirt and oil from surfaces and skin. To check your understanding, how do you differentiate between a saponifiable and non-saponifiable lipid? That's right. Saponifiable lipids have the ester bonds which can be hydrolyzed, and non-saponifiable lipids don't have those ester bonds. Therefore, they cannot be hydrolyzed. Examples of non-saponifiable lipids are, yes, steroids. They don't have the ester bonds that we just talked about. Therefore, they are non-saponifiable. Finally, let's connect all this lipid chemistry to something very relevant to our daily lives: lipids in our health and nutrition. While lipids are incredibly valuable for energy storage and essential cellular functions, excessive intake is linked to significant health risks. One major concern is atherosclerosis. This is where cholesterol, particularly low-density lipoproteins, can deposit on artery walls, forming plaques that narrow arteries and increase blood pressure. When these arteries become severely blocked, it can lead to serious events like heart attacks or strokes. This is why dietary recommendations are so important for reducing cardiovascular risk. We're often advised to eat less red meat, increase our vegetable intake, and consume more fish. These choices help lower lipid and cholesterol intake, reducing excess fat storage and cardiovascular strain. However, it's crucial to remember that cholesterol isn't all bad. It has essential roles. It's vital for hormone and vitamin D production, it insulates our nerves, helps synthesize bile for digestion, and it is a key component of cell wall structure. In fact, our liver naturally makes all the cholesterol our body needs. The body transports cholesterol through the bloodstream using packages called lipoproteins. These have different types. We have HDL, often called good cholesterol or high-density lipoproteins, which removes cholesterol from arteries and brings it back to the liver for disposal. Then there's LDL, low-density lipoproteins or bad cholesterol, which deposits cholesterol in arteries, contributing to plaque formation. Alright, to check whether you have retained info, I will flash a series of molecules. Tell me what classification they are based on their structures. Ready? Let's go! Wow, you did a fantastic job. That brings us to the end of our deep dive into lipids. I hope you've gained clear understanding of these vital biomolecules. Attached below is an Anki deck containing structures of molecules of lipids. Input below the time it took for you to answer correctly all the flashcards. Compete with others to this minigame and you may just win this review center's grand prize. Also attached in this video is a step-by-step process of a test for unsaturation. You can see the cool differences between the saturated and unsaturated fatty acids. Before you answer this review center's mock exam on the lipids board exam, is there any question you want to ask or anything you'd like me to clarify before you move on to the next video? Just comment your question below and surely we will get back right to you.